Professor Annette, moving on to you. You gave a presentation on lipid-based system, SNATs in particular. So could you start by explaining the rationale behind using this system as a solubilization approach?
Well, the rationale is that it's, in a way, it's a relative simple formulation. It's a mixture of triglycerides and hydrophilic surfactants and maybe a co-solvent. And they are mixed in a ratio so they give a oil-like liquid. And that oil-like liquid you can easily fill into a soft gel or a hard gel capsule for that matter, and then it's basically ready as a dosage form. Then what happens once it's ingested is that in the gastric fluid it will disperse and form nanoemulsion particles or emulsion particles. And those particles or droplets, they will contain the drug in solution. So in that way you have delivered a poorly soluble drug in solution to the gastrointestinal tract. It's not in solution directly in the gastric or intestinal fluids, but it's in solution inside that lipid particle, and that facilitates the partitioning into the intestinal fluid and then subsequent absorption.
Right. So say, for example, someone is interested in developing a lipid-based drug delivery system. What would you say are the key considerations? What are the requirements? What factors should they take into account?
Well, they need to consider first of all the stability of the drug in the lipids. Because if the drug is not stable at all in lipids, then you know that you're getting a problem in the stability of your dosage forms and so on. Then, of course, if your drug is unstable in the lipid, then there's a lot of other tricks you can do by, like having the drug in a suspension or co-dosing it with a lipid, and that will still increase the bioavailability, but it's getting more complicated. If your drug is stable in the lipid, then you want to assess the solubility of the drug in different excipients, and then you want to select the excipient that can give you a self-emulsifying system while keeping the drug in solution. That's sort of like the traditional way. If then you want to reduce the excipient load, the number of capsules, the volume of lipid that you have to ingest, if that's relevant because the solubility of your drug is still relatively low compared to the dose, then you should go and consider, can I supersaturate the drug? Like I explained about yesterday, some drugs are very good at supersaturating in lipids, and that way you can actually have a higher dose of the drug in a lower volume of lipid.